Christina just said, I'm current postdoc at the Prince William Sound Science Center, focusing on mariculture. So today I'll be talking about my work helping out farmers by growing kelp in the commercial hatchery, as well as answering some very industry relevant questions with my research. But first, before we begin, let's take a step back for a minute and talk about some kelp basics. Oh, I need to use this, I forgot. So this mini KELP 101 session is going to take a Q&A form. So if you do know the answers, feel free to shout them out. So question number 1, is kelp a plant? No, but John knows this one. So seaweeds are not plants. So green algae are the most closely related to plants, but I messed up some of my animations here, but that's fine. But what we've got here is we have our green algae pretty closely related to green plants, but then red and brown algae are actually further along the evolutionary tree. So they're pretty distantly related. And then up here we have us. So pretty distantly related. You can see all of those. So even within the realm of seaweeds, there's a lot of evolutionary diversity. So question number 2, what is the difference between an algae, seaweed, and kelp?
Kelp is a type of seaweed. Brown seaweed.
Yes, it is. Christina got that. So kelp is a type of seaweed. So if we consider, that went too fast. If we consider algae being a really big bubble, within algae, you can kind of split it into 2 different groups. You can split it into microalgae and macroalgae. So macroalgae are what we think of as seaweeds. So what we can see is that all seaweeds are algae, but not all algae are seaweeds. Then within each of these, I don't know if this is like an automatic timer, and I don't know why these are moving so fast, but that's okay. Within these groups, we can see that like within the group of macroalgae and microalgae, actually, we get 3 different groups. So we get green algae, we get red algae, and we get brown algae. And so then actually within the brown algae, we get a subgroup called kelp. And so what we find out is that all kelp are seaweeds, but not all seaweeds are kelp. I'm going to pause for a second and figure out what's going on with these animations, because this should not be going automatically. Give me 1 second, y'all. Yep. Maybe going on click, unless it's just really, it could just be really, really sensitive. That's fine. Okay, we'll try again. And I'll try and get it to stop going. But that's okay. It'll be fine.
What do you mean you're not talking at 1,000?
Yeah, exactly.
Words per second.
It's all good.
Let's drag this out. Oh, it's still doing it.
Yeah. Hopefully it's just this slide. If not, we're just going to be going back and forth a lot throughout the presentation, which is what it is. Okay, sorry everyone. Okay, so pausing here. What makes red, green, and brown algae different? Anybody have any ideas?
The colors.
Color. What I heard something over here.
Pigments.
Pigments. Got it right. So the pigments are the main thing that kind of differentiate these red, green, and brown algae. As I showed you earlier, we can see like there is also a lot of like evolutionary difference. There's a lot of like reproductive difference between these different types, but pigments are kind of the main factor that differentiates these from each other. And so we can kind of see like different pigments will absorb different wavelengths of light, and then different pigments are represented by different algae. So green algae is represented by chlorophyll B, neoxanthin. Brown algae is chlorophyll C. Sorry, let me go back. There we go. Fucoxanthin, and red algae is phycoerythrin and phycocyanin. But yeah, pigments generally make up the difference between all of these. It's all good. I think it's just more sensitive than I was giving it credit. Okay. So question number, I think this is 4 now. Do kelp have seeds? I'm seeing Becca nod yes. I also saw a no. So what we've got here now is the answer is no. Kelps do not have seeds. So what do kelps have instead? So let's go through the life cycle of kelp really quick. So what we start with is macroscopic stages. So that's a diploid stage. And then we also have some microscopic stages on the other side. Let me just see if not holding. We'll let it fill in and then we'll go through it. So
Once we're taking over.
yeah, exactly. All right, so what we have is we start with macroscopic stages on the left side of the reproductive system, and then microscopic stages on the right side of the reproductive system. So the macroscopic stage is what we call a sporophyte. So this is diploid, meaning that it has two sets of chromosomes. This is the main seaweed that we'll see out in the ocean. When that particular individual gets reproductive, it'll start to generate this tissue called the sorus. And so that's essentially like a cluster of a bunch of spore creating cells and everything. So then the sori will then release spores into the water, which will settle into the benthos, into male and female gametophytes. These male and female gametophytes are a completely different life stage than the sporophyte that we start with. They're actually they're haploid, which means they only have one set of chromosomes, and so those will actually undergo sexual reproduction, fertilize each other, and then we get a new female gametophyte producing an egg, which then turns into a little baby sporophyte to complete the cycle. That's how kelp reproduces. It's through spores, not seeds. Last question. How does kelp farming work? I won't wait for any answers because this is a multifaceted question. I'll just go straight into the answers myself. Well, so there's a few different ways. So kelp mariculture in Alaska takes place in a few different regions. So right now there's kind of four main regions that are represented in the kelp mariculture world. There's Kodiak, there's Homer and Kachemak Bay, Cordova and the Prince William Sound, as well as Southeast Alaska. So I just kind of lumped Juneau, Sitka, and Ketchikan together down there. But kelp mariculture in Alaska right now, a lot of people are focused on growing mostly three main seaweeds. Sugar kelp is the most popularly grown seaweed. It's what has already a really big market. And then it's followed by ribbon kelp, as well as bull kelp. And so I do a lot of research with all three of these species, but these are kind of the three main species of interest in Alaska. So we already talked about the life cycle just now. So how does the life cycle then translate into kelp farming? Well, it starts with, again, the animations are running away from me, so I apologize. But we start with wild kelp bed. So what will happen is farmers will go out and collect from that wild kelp bed, bring them back to the hatchery, and then the hatchery will cultivate seed string, which farmers will then eventually take and plant out onto the line. So in the hatchery, we kind of see we see all the really like microscopic stages growing, and then once it becomes macroscopic and turns into that diploid stage that we talked about earlier, it goes out onto the farm and is eventually harvested, and then the cycle closes there instead of continuing. So when does this happen during the year? Well, most of the times farmers will collect sori between August and September, bring it into the hatchery, which is when my job comes in. We grow it for about 2 to 3 months, just depending on the species. And then the farmers will take it and outplant it, and the farm season will run from roughly like December to May when it will be harvested. So that brings me into part 2, the commercial hatchery. So the Prince William Sound Commercial Hatchery. Why do we have a commercial hatchery at the Prince William Sound Science Center? Well, we were originally developed to solve the problem of kelp farmers in Cordova having few hatcheries in the state that they could source seed from, which meant that they had to do a lot of shipping back and forth between here and places like Juneau or Kodiak or places that already had hatcheries. So our solution was to establish a local hatchery to provide quality seed strain to local kelp farmers and potentially reduce the cost of getting that seed strain. So the commercial kelp hatchery here has been operating since 2023. It was begun by Alicia Cipher, and she's been our hatchery manager. And this past year, we made a lot of recent improvements and upgrades, which I'm going to tell you a little bit about today. But those are in 2024. For those upgrades, Alicia received an equipment grant from the Southeast Conference through Build Back Better mariculture funds. And that we got a lot of new hatchery infrastructure installed this year. And then this past fall, we also hosted the hatchery operators training workshop, actually in this room right here, which was run and operated by Greenwave. So one of the main infrastructure upgrades that we use this year to kind of make our kelp farming process better is temperature control. So if you've been into our wet lab space at all recently, you might have seen this giant silver box in the corner. And that's basically where a kelp hatchery is housed. So essentially what the Silver Box is, it's a walk-in refrigerator. So you walk in and you can like control the temperature as much as you want. So during the commercial hatchery season, we kept the temperature at roughly between 10 and 11 degrees for the entire time we had kelp in. And now that I'm running some experience, I'm able to lower the temperature as low as I need. So right now it's at about 5 degrees Celsius. And so on the right is what the inside of this container looks like. So you're able to walk in and we have a bunch of seawater tanks set up inside there where we're then growing our kelp. Another infrastructure upgrade we had this year is that we had massive improvements to our water filtration system. So one of the issues we've been having with seed previously is that there was a lot of contamination. We get gross stuff like green algae, diatoms, all that other stuff, really messing up the kelp that we were growing. So we wanted to make sure that like our water was staying as clean as possible. That way our kelp grew really nice and healthy. Really important for delivering good product to our farmers. And so our water filtration system right now does a bunch of different stuff. We start by bringing in water from the sound about 100 meters outside the science center right here. We pump it into the lab. It will go into a skimming tank, which then passes through an ozonator. So we're basically pumping the water full of ozone, and that's meant to kind of kill everything that's in the tank. After it goes through that for a bit of time, then we pass it through a series of filters, which you see as like the blue cylinders, just to pull out any little particles or little baby organisms that are still in the water. And then we bring it into the hatchery, and then it passes through a UV system. So then a UV light bulb will also do the work of killing anything that's even left over. So it's going through a lot of steps basically to make sure we're getting the cleanest water possible in our hatchery system. And then we also have a bunch of other new equipment this year. And so I mentioned we go through at least one UV filter before it gets in. Pardon the picture of the dirty tank. I didn't have time to clean it before I took this photo today. But each tank is also equipped with an air pump system, which is the kind of tubing line that you see. So that will kind of like keep the water moving and flowing. Even if we don't have water actually coming in. Each tank also has its own valve where we're able to fill the water and exchange the water in the tank as we need individually. And then that little black thing on the side is actually another UV filter. So each tank, like while the water is in the tank, it's also being circulated through a UV filter to kill anything gross that's growing as well. And then we also got really good fancy new microscope this year, which allows us to do much more rigorous checking of our spools as they're growing. And this is an example of just like how good of quality we can kind of like look at like little baby kelps as they're growing on our line and stuff. So we have a lot of really cool infrastructure this year to make our hatchery even better at serving the community. And then we've also done a lot of protocol upgrades. So I mentioned that we hosted the Green Wave Hatchery Operators Workshop this past year. And the goal of this workshop was to increase kelp quality throughout the state and provide knowledge exchange among different hatcheries. So we hosted, when I say we hosted the workshop, we provided the space for everybody to come here and sit and learn together, but Green Wave were the ones who were bringing all the knowledge, like all the expertise to the workshops. At this workshop, 8 Alaskan and 2 East Coast hatcheries were represented, where we were all able to chat together about our issues, and do hands-on demonstration of protocols, and discuss common hatchery issues such as water quality, sanitization, contamination, communication with farmers, and general business approaches, which have all been barriers to producing good kelp seed in the past. And then each hatchery was able then to bring the takeaways from each topic into our hatchery protocols. And I personally was able to attend this, and I learned a lot, and it definitely made our hatchery season really good. So that's kind of all the changes we made to our hatchery this year, but how did our 2025 production go? Well, it went really well. We produced over 52,000 feet of seed string this past fall, mostly sugar kelp and a couple different batches, but we also did small batches of bull kelp, ribbon kelp, as well as giant kelp. So we grew at least 4 different species this year, and there's interest in growing a lot more species of kelp that can be found around Prince William Sound. And overall, like 95% of our schools were high quality. So what this means is all those infrastructure and protocol upgrades that we just made really paid off. Over here, you can kind of see a picture of our worst, average, and best spools. And you can kind of see that all these spools look pretty good, even our worst one. That one's only the worst just because it has some like weird patches that doesn't have that much kelp, but overall, all the kelp is looking nice and brown and really healthy And so all the farmers got really good seed to take out onto their farm. As I mentioned, we produced great kelp and very happy farmers, which was very exciting this year. These infrastructure upgrades paid off. Our abiotic variables were steady. I was monitoring them daily, saw very little change, which meant our really cool temperature control system was working. And our water filtration system was also fantastic. We had very little contamination. We just needed to change our filters a bit more often. Yeah, and we also were able to put together a lot of protocols so that we can keep doing this good work into future years. And so, I mean, we're still working on some stuff, right? Like I said, like we saw, there was some patchy kelp. There's still a lot we can still improve on, even despite our success. And so some of our continuing challenges and future directions is we really learned that running a hatchery is a lot of labor. For the amount of kelp we produced, it was a lot of work. And so we're definitely hoping to hire a technician for future seasons. And we're also working to formalize some of our business practices just to make the communications with farmers and agreements a little bit more streamlined. We're also working to better understand kelp species that are currently not commercially cultivated. And we're also trialing gametophyte seeding techniques, which brings me into the next part of my talk. Hatchery experiments. So for this part of my talk, I'm going to be focusing kind of on two main themes of what I do for research. So my research right now is primarily focused on gametophyte seeding techniques, which I'll get into in a minute, as well as better understanding the conditions that we're growing our kelp under. So research focus number one, what is needed to incorporate gametophyte seeding into kelp hatcheries? Well, let's take it back up and let's understand what gametophyte seeding is first. So I already went through the life cycle with you. So we have our sporophytes, which release spores that then turn into gametophytes once they're on the bottom of the water. So what is gametophyte seeding? Well, the typical way that spools are normally seeded is what we'll do is we'll take the source from our adult tissue, we'll get the spores out of it, and then we'll basically create a spore solution. So we have all of these spores suspended in water, and then we'll directly dunk our seed spool into that water so that the spores stick to the seed string. So what gametophyte seeding is we then take instead of putting those directly onto their spools, what we'll do is we'll allow the spores to develop into gametophytes. We'll allow the spores to develop into gametophytes in cultures on their own. And then once they've already developed and gotten a bit older and more mature, we'll then basically paint them onto the spools with a paintbrush. And so there's a few different possibilities that come from this. And I'll get into like the benefits of gametophyte seeding in a minute. But we're kind of experimenting right now with some of these different options for actually seeding our spools. And so right now, one of our questions is, how do spools seeded from gametophyte culture compare to those seeded from spore culture? And then, like I mentioned, gametophyte seeding has a lot of advantages. We might have more control over the density on our spools. We might have more control over the timing of reproduction. If kelp is only reproductive at a certain amount of time each year, if we're able to keep gametophytes and keep them for a longer amount of time, we can start our spools whenever the farmers are ready without having to rely more on natural cycles. There's also a possibility of reduced contamination in our cultures. And it also allows us to reduce our reliance on wild blood. That's where if we're able to kind of build a gametophyte seed bank, we may not have to go out and take as much wild material from the environment every year, which is less disturbing for the environment and keeps environmental conditions more intact. So before I get a little bit more into my research on gametophyte seeding, I also need to kind of give you a little bit of background in Alaskan mariculture policy. So right now, the way that fertile kelp is brought into the hatchery, we have to follow what's called the 50-50 rule. So the 50-50 rule essentially states that to mitigate genetic fitness risk and maintain genetic diversity, seed stock must be sourced from more than 50 unrelated parent plants, which must be collected from within 50 kilometers by water of the outplanting site. So 50 parents from within 50 kilometers. That's the 50-50 rule. However, the 50-50 rule also makes the assumption that all wild broodstock individuals collected are equally likely to produce offspring. However, there's not really any data to back this up. Current spore seeding methods do not rely on proof that all sori are releasing myospores during the seeding of spools. But asking hatcheries to make sure that we are actually getting 50 parents to contribute to the schools that are being outplanted is actually it's a really big ask on hatchery managers, and it would be very unsustainable as a business practice. And could also potentially result in overharvest of wild populations, where if you get a batch of kelp that's not all fertile, then you have to keep going out again and again and again until you get 50 fertile kelp individuals. So what we're kind of asking right now is whether gametified culture could be better at ensuring the goals of a 50-50 rule than spore culture. So what we're doing to do that is we are currently running a project that seeks to quantify the effect of 3 different seeding methods on genetic recombination and seed quality. So essentially we're using traditional seeding with just spore solution. We're seeding with a mixed parent gametophyte solution. So essentially we're taking that spore solution and just letting it become its own gametophyte culture. And then we're also seeding with what's called a controlled parent solution. So we had 50 parents, created 50 different gametophyte cultures from each parent, and then combined those cultures together to make sure that each parent's offspring were represented equally in what we're using to seed our schools. And so the goals of this project are to clarify whether alternative seeding methods might better preserve the genetic diversity of Alaskan kelps, and clarify whether alternative seeding methods have the potential to increase the quality of cultured seaweeds. In addition to preserving the genetic stuff, are we also seeing better kelp grow on our schools? And I'm working on this project with my collaborator, Dr. Felipe Alberto at the University of Wisconsin-Milwaukee. So we were running this is, as I mentioned, we have our 3 different seeding trials. We have spore seeded, gametophyte seeded from that spore solution culture, and gametophyte seeded from 50 different parent gametophyte cultures. And we're doing this with parents from Cordova as well as Kodiak. So we have kind of 6 different combinations of this. Once we grow them in the hatchery, we're then going to put them on 2 different farms in Cordova and Kodiak on a spreader bar array that kind of looks like this. So each of the different colors on the line just kind of represents a different treatment, but essentially it's a 2-line array that's going out onto the kelp farms so that we can plant out our kelp and see how it works. There we go. And from this, we're going to be collecting a lot of data. So we're collecting genetic data at 3 time points. We're getting genetic data from the parents that we're using to create these cultures. We're getting genetics from the offspring that leave the hatchery, and then we're getting genetic data from the offspring that are harvested in May. We're also getting seed quality data, so how long it takes for gametophytes to become fertile, what does spool coverage look like, what does spool color look like, how big are the sporophytes when we outplant them. And then we're also getting harvest quality data. So how does this affect yield and density and biomass when we harvest this off the farm sites? So currently, where are we at with this project? We just outplanted our Kodiak spools. They're in the water right now, and we've got our juvenile sporophyte genetic data already. Our Cordova spools had a little bit of a delay. We had an initial massive contamination issue, so we had to restart some of our spools. So they're actually actively in our hatchery right now growing, and we're hoping to get them out in 2 weeks after the mariculture conference, which is taking place next week. And then we're also still maintaining all of our non-contaminated gametophyte cultures, just in case we would like to use them for future experiments. So our takeaways so far, how is gametophyte culture comparing to spore seeding culture? Well, I found that cleaning methods really matter. I did have more contamination issues than I expected, but I used a very different, sorry, it's doing the automatic switch again, but I did also use different cleaning methods than I would have for my commercial hatchery. And so I'm definitely gonna be switching that in the future. I also found that maintaining individual parent gametophyte cultures is very time intensive. I had duplicates of each of my 50 individuals from 2 sites with 2 duplicates each gave me 200 gametophyte cultures to maintain, and that would take me about it took me 18 hours to release 50 sori on the same day, and then it generally takes me 3 to 4 full work days to check the health of all of these cultures. I was also the only person working on this project, so I didn't really have a lot of other help at the same time. And so my main takeaway and what I'm going to be approaching the state with is that starting gametophyte cultures the same year you need to outplant is likely not a feasible strategy for commercial kelp hatcheries, which kind of brings us into a question of 100% replenishment of seed stock. Right now, there's this assumption within Alaska that you need to have 100% new seed stock anytime you're starting to bring kelp into the hatchery. That's not actually a requirement. Sorry, I'm just going to go through all of them and pause it. So, okay, so it's not actually a requirement. There's not really distinct guidance from ADFG. It's still reviewed on a case-by-case basis, but they don't really have anything to say like, oh, like this is how much new fertile material you need to bring in every year to make sure that you're maintaining genetic diversity of what you're outplanting. So we're trying to now start asking the questions, how does using communified cultures from previous years affect the genetic diversity of the seed line? And how does the genetic diversity of communified cultures change over time after fragmentation? And so we're actively looking for funding right now to extend this project to answer some more of these questions. So what we're planning on doing this year is we're planning to rerun our initial experiment and add 2 more treatments. So we're trying to add in gametophyte seeded treatment from 2025 to compare to our 2026 trials. And we're also trying to combine gametophytes from last year and this coming fall to see whether one year is represented more actively in another. Like maybe there's a benefit to being an older gametophyte, maybe there's a benefit to being a younger gametophyte. And so if we do a mix and kind of see how the different parent sources are represented in our offspring samples, that'll be really helpful and tell us a lot. And then we're also just looking at, how fragmentation, so as we split cultures and replicate them over time, how does that affect the genetic diversity within those cultures? So that takes me away from genetics and more to research focus number 2. What are the best conditions for running an Alaskan kelp hatchery? So right now, there's a lot of study going on bulk kelp temperature tolerances. Back in 2016 or 2013 to 2016, the warm blob hit the west coast of North America, and it negatively affected a lot of bulk kelp populations along the west coast. Down in California, they saw up to 90% losses of their bulk kelp stands, and as you go further north, that percentage decreases a bit. In Alaska, we got hit a bit, but we didn't get hit so bad that there were suddenly like massive losses of our bulk kelp stands. So right now, a lot of the studies of temperature on bulk kelp are really focused on these higher temperatures, figuring out like when growth and physiology is best, when does mortality start. And generally we see that bulk kelp can grow like up to 18 degrees Celsius before it really starts to die. However, people haven't really been looking at low temperatures very much. There's one study that shows that low temperatures roughly double the time to egg production. But what about when the kelp reaches the ocean? In Alaska, if you look at the monthly average coastal water temperatures, we actually have pretty low temperatures. From October through March, most of the time, the temperatures are anywhere between 2 and 8 degrees Celsius, which is a lot lower than what people are studying right now. Currently, hatchery conditions generally aim to culture kelp at about 11 degrees Celsius, which is a little bit higher than the water they're actually going into. So do we actually need to be culturing kelp at temperatures that high, or should we be culturing our kelp at lower temperatures before it goes in the water? So the next study I'm going to tell you about is we're working to optimize kelp hatchery production and farm harvest by understanding the lifelong effects of abiotic conditions. So we're essentially assessing how varying temperature and light levels influence microscopic bull kelp growth and development at the hatchery stage. And we're also determining how the growth and harvest of adult bull kelp is affected by different hatchery conditions. So this is a little bit of a complicated schematic, but essentially the takeaway is that we were doing this in both Kodiak and Juneau. And this is kind of a carryover from my past position, which is why Cordova is not included in this study. But in Kodiak and Juneau, what we did is we essentially had 4 different light treatments and then 3 different temperatures. And we were essentially looking at how lower temperatures from 5 to 8 to 11 degrees Celsius. And again, 11 degrees Celsius is kind of the standard for kelp cultivation at the moment. How do these lower temperatures compare to what like the current standard for kelp cultivation? And then also like, do we need lower light levels? Do we need a little bit higher levels? Should we be changing the light levels as the hatchery is taking place? And so we did this.
Take into account light color.
Light color. We did not take into account light color. We're just kind of generally using bright white LED lights. That's a good question. People have done studies on light color before though, and I can refer you to those. And then essentially though, like once we grow this in the hatchery, then we're again outplanting it at 2 different farms, Sequester Farms in Juneau and Kelp Island, Alaska in Kodiak. So how did this go? So we started this project last year and then we're still working on it. But essentially we got mixed reviews based on our hatchery results. Kodiak had successful gametophyte growth in 2024 in all treatments, but there was a lot of variation in the quality of kelp spools produced. As you can see, like some of them look kind of healthy, some of them look very, very sparse. For some reason, we had a little bit of a seed plant failure in our 8 degrees Celsius treatment. Nobody really knows why, but the spores just weren't sticking. So those were just overall generally lower. And then our 11 degrees Celsius treatment, right now, it doesn't look like you can see any like little baby kelps. There were baby kelps there. We actually kept them in the hatchery too long. And so all the little baby kelps fell out off before we put them in the water. So it's a little bit chaotic. Juno had successful settlement, but for some reason the gametophytes never germinated. So all these little puffballs are just really large gametophytes, but none of them really started producing those little baby kelp sporophytes that we want to see before we're actually putting our kelp out on the farm. So it's kind of weird. But we took them and we did some data on what we were able to see, and what we saw was that Juno generally had higher spool coverage at higher light, regardless of temperature. And Kodiak spool coverage depended a little bit more on temperature than light. Again, there was a settlement issue in 8 degrees, but we had almost 100% coverage under 11 degrees Celsius and a little bit lower percentage of coverage under 5 degrees Celsius. Additionally, we saw an effect of gametophyte lengths. So again, we saw a little difference between light treatments for Kodiak, but we did see that increased temperature faster growth. So the green box, the teal boxes are week 4, the blue boxes are week 2. Sorry, these animations are just really messing with me today. But essentially what we saw here is that, generally the warmer temperatures had the longer gametophyte lengths, and then the difference between week 2 and week 4 was also greater for the higher temperatures than the lower temperatures. So higher temperatures were resulting in faster growth for our babies. So how did the harvest go? So despite the issues we saw in the hatchery, we still were like, well, let's put them out on the farm and see what happens. Again, the results were not great. Juno didn't see any kelp grow at all. Kodiak had very sparse kelp coverage, but we were able to harvest 42 kilograms of kelp, and so we tried to collect some data on that. And what we saw from Kodiak's harvest is that kelp grown at 5 degrees Celsius had significantly higher density and yield than kelp grown at 8 or 11 degrees Celsius. Again, this was kind of weird. Like when we were looking at the gametophyte results, we saw that 11 degrees Celsius was actually better for kelp growth in the hatchery. But then once they were on the farm, it seems like the 5 degrees Celsius might be better for growth. And so we started to ask, why did the 8 and 11 degrees Celsius treatments have such poor density and yields? And there's a few different options we're looking at right now. So one hypothesis is that last year was a really weird winter. I think a lot of it that was true for Cordova, that was true for Kodiak, that was kind of true for the entire state of Alaska. In Kodiak, it was one of the cloudiest years on record, and we got almost no snow the entire winter. And so it was dark, it wasn't as cold as it usually was, and there was a lot more freshwater input with all the rain we were getting as opposed to snow. So we're starting to look at whether the environmental conditions might have an impact on how bull kelp grows when you outplant it. Our second hypothesis is that hatchery factors such as too frequent germanium dioxide additions, which is used to control diatom contamination, and too low water motion may have reduced the attachment strength of the kelp we outplanted. To answer this, we did a experiment looking at 3 different germanium dioxide frequencies of inoculation and 3 different water motion levels. And generally what we saw is that there were not really any significant differences. You can use germanium dioxide however often you want. It's not really going to affect how well the kelp attaches to the seed line or how big it grows. And the same for water motion. It's good to have water motion, but there's not really a huge difference between how high the water motion is. So our third hypothesis is that the 5 degrees Celsius kelp actually did grow better in Kodiak. And so what we're doing right now is we're currently rerunning the experiment. So I got kelp from Kodiak shipped up to Cordova this year. We're rerunning this experiment with our 5 degrees Celsius treatments. We dropped our dynamic light treatment, so we're just looking at low versus high light. And we seeded our spools with gametophytes this time versus spores. And this is I won't go into that. You can ask me later. But we're getting more data again, and it's still in progress. We were hoping to outplant this week, but we're going to wait another couple weeks before we outplant. But it is almost ready to go out. I was looking at all of it under the microscope, and I am seeing all the little babies on my 5-degree treatments, which means it's ready to go. So our preliminary results for 2026 is, again, we're seeing a positive effect. We're seeing little difference in percent cover after 2 weeks of growth. So again, those dark blue treatments, they're all kind of on the same line, showing more or less like the same amount of percent cover. But then after 4 weeks of growth, higher temperatures generally do seem like they're leading to higher percent cover among our treatments. And so again, if we look at gametified lengths this time, in 2025, just as a reminder, in 2025, gametified lengths did increase faster under higher temperatures, and we're roughly seeing the same pattern, just a little bit more variance in the data this year. So again, we're starting to see a little bit of an increase between week 2 and week 4 among higher temperature treatments, but it just kind of depends. Also, I am, again, really sorry about this runaway train of slides that I had going. But anyway, so our preliminary takeaways right now is that low versus high light doesn't really have a huge difference on growth. Low light might be better for kelp growth, but we have a lot more data to analyze. And in terms of low versus high temperatures definitely lead to faster growth in the hatchery. But the effect of hatchery temperatures on performance once outplanted is still inconclusive. So we're waiting on those results right now. But it's possible. Lower temperatures may lead to better performance of outplanted kelp. And so we're currently at 36 minutes. So I'm actually going to pause for a moment, this runaway train, and kind of just, actually, no, we got time for this. And so part 4 of my talk is just about the kelp farming community. So in addition to all the work I do in the hatchery, both growing commercially and experimentally, I'm also working a lot with the kelp farming community in Alaska on a variety of different projects. So the main thing I'm involved in right now, in addition to my role as a researcher, I'm also the co-coordinator of something called the Bull Kelp Research Squad, aka Beakers. So we were founded in 2023. Some kelp industry members and researchers approached Alex LaFerriere, who's a scientist at NOAA, to see if she would be able to help them facilitate some discussions. Essentially, before 2023, a lot of people were trying to grow bull kelp in Alaska, but very few people were actually making progress. Everybody was kind of working in a silo. Nobody was really talking to each other. So it was really hard to try and move the industry forward. So I was starting my Sea Grant Fellowship with Alex at the time. And so she approached me, asked me if I wanted to be a part of it. And since then, we've kind of been working together as the co-coordinators of Beakers. But the goal of Beakers is to develop a current understanding of how we're growing bull kelp in Alaska, what is and is not working, with the end goal of developing a proof of concept for how to grow bull kelp and make it scalable. So the way we do this is we foster discussions around the successes and challenges of growing bull kelp, and we develop resources for those growing bull kelp commercially and experimentally. So right now, Beakers consists of 30 members. We've got a variety of different kelp farmers represented within our group. We've got several different industry members, such as Greenwave and Barnacle Foods. And we also have a bunch of researchers at University of Alaska Southeast, UAF, and then obviously here. Hello. And we also have a few agency members represented as well, including the Alaska Fisheries Science Center at NOAA and Sea Grant Alaska. Right now, Beakers is primarily focused on work in Juneau and Sitka, Kodiak, and Cordova. And since we've started, there have been over 15 plus research projects ongoing, just studying bull kelp and how it grows in Alaska. These projects are studying hatchery topics such as seed string innovation. So how do we use less plastic-based seed strings? Are there any natural alternatives we could use? What is the efficacy of different fertilizers? Are there cheaper options in the industry standards? What are the optimal abiotic conditions? I just told you a little bit about some of the work we're doing on that. There's also a lot of farm topics. So what is the appropriate outplanting depth? How deep in the water column do you need to put your bull kelp? How does seed density and line spacing affect how bull kelp grows? Should we have bull kelp closer together, farther away? And it's farther away because it will tangle very easily. And then how do we also prevent buoyancy? Unlike other kelps, bull kelp is basically a balloon. Floats to the top of the water. So it's not going to, as it grows and gets bigger, it's actually getting more floaty instead of heavier. So how do you combat the fact that it keeps wanting to float up to the surface and keep all your lines submerged on the farm? There's also a lot of innovation and array design coming out of all of this work. And there's a lot more other projects. As I was telling you earlier, I'm also doing some work on KELP-ING genetics. And then there's a bunch of other projects that are also ongoing looking at help in the state. So our research priorities for the year, sorry that I messed up these animations. Every year, one of the things Beakers does is we also vote on our research priorities and topics for the coming year. So we just had our vote actually last week about our research priorities for this coming farming season that's beginning this fall and going through early 2027. Essentially, our priority topics for this year are better understanding the timing of hatchery and farm operations. When is bull kelp fertile in different areas? When should we be getting it into the hatchery? When do we need to outplant it? And can we outplant it more than once over the course of the year? Farm engineering, so buoyancy is always an issue. How are we designing farms? Is there an optimal way to design farms? And then also genetics, like how do we better understand bull kelp genetics in this state? And so some of the questions that are coming out of this are, what is the effect of outplant timing on yield? Can we stagger outplanting? How do we space seed in a way that reduces hatchery capacity needs and reduce costs for farmers? Are there alternative seeding techniques we can test, such as gels or spray-on techniques? Why are bull kelp communophytes sometimes staying vegetative? I already mentioned, I told you about that in one of our projects, is for some reason that one batch of bull kelp was just not getting fertile at any point. And then we're also starting to expand our interest into other buoyant kelp, such as giant kelp. So for macrocystis, when is macrocystis fertile? And macrocystis is giant kelp, by the way, for those of you who didn't know that. And then also, does seeding density matter for giant kelp if it self-thins, and how do you think about density on those lines? We also now have a website, so if you scan this QR code, you can visit it. But find us at bullkelpresearch.org. We're trying to keep this up to date, and this is going to be where we keep all of our resources as we come out of them. And then we also, every year, starting last year, we've also started hosting bull kelp workshops. So last year we had people represented all along the coast of the west coast of where bull kelp grows. So the dark green line along the west coast kind of shows the range of bull kelp, and the stars are just where we had people coming from to join our workshop. And then we had that again this year, and we actually added a lot more participants throughout the coastal US to better understand the work being done, to better understand how we're growing bull kelp. And the report and takeaways for that are coming soon, and those will be available on our website when it comes time. And so what is the future of Beakers and bull kelp farming? Currently, we're working on our new projects for the 2025 to 26 harvest season, and we've already been seeing a lot of amazing progress in bull kelp being farmed. One farm in particular, Sequester Farms in Juneau, is kind of the gold standard for how to grow bull kelp. They've successfully grown it for 2 to 3 years now, and they've definitely have it almost down and unlock, like how to successfully grow bull kelp on a farm. This summer we'll be drafting a bull kelp guide based on our experiences growing bull kelp, and so this is kind of like meant to be a working document bringing everybody's experiences together about how do we think about bull kelp in the hatchery, how do we think about bull kelp on the farm, what do we do with it once we have it. And then moving forward, we're expanding our scope to include more buoyant kelps, and we're increasing conversations with people restoring bull kelp further south. And with that, that is the end of my talk, and I'm happy to take any questions about hatcheries, research, bull kelp, kelp in general, seaweed, all the things. Thank you. And back away from the runaway train. Yes.
Do you have any collaboration with the Monterey Bay Aquarium and their or something?
Currently, we don't have any collaboration with Monterey Bay. Right now, a lot of the people in California, they're very much focused on kelp conservation and restoration, which is like aligned. We're, like I mentioned in my last slide, we're starting to increase those conversations with people down south who are focused more on conservation and restoration, just because we think we can probably learn from each other in the ways that we're growing kelp for our different purposes, commercial versus conservation. But yeah, we don't currently have any, but I'm planning on reaching out to them at some point in the next year. Yes. Yes, Scott.
Where are you sourcing your macrocystis for Prince William Sound?
Prince William Sound, there is apparently one very tiny bed right out on Montague Island. That's where we got ours from. It is outside the 50-kilometer permit or the 50-kilometer range, but that is one of those things where if you make a good case to ADFG, they can give you an exception to go outside that range. So we got permitted to get some giant kelp from outside our range this year.
I have a question
Yes.
online. I put it in the chat. Can you
Oh,
see it, or should I just ask it?
I would prefer if you asked that. That would be amazing.
First of
I'm
all,
working on
thank
2 different
you.
computers right now. Yeah.
Thank you, Angela. I really appreciate your talk. I wanted to know is how are you providing these kelp spools to the kelp farmers? Are they getting them free of charge, or is there something other to help with the cost recovery?
Yeah, so we are essentially charging farmers for the cost of production. So right now, spools are costing about 50 cents a spool, or 50 cents per foot, rather. So each spool itself is about 400 feet, and so each spool is probably about $200. And that primarily covers the cost of production. So it takes a lot of electricity to run the hatchery. We use a lot of materials during that time, a lot of like nutrients we have to supplement. And all that stuff. But it's basically just the cost of production. We're not really making a profit off of that at the moment.
Great. So a follow-up on that, if they don't get it from you, where are they sourcing kelp from?
There are a few other hatcheries in the state who are also able to produce kelp. So I think I mentioned really early in my talk, there are currently 8 Alaskan kelp hatcheries that I know of. Native Conservancy in Cordova, they did just start one. I'm not sure if they produced any kelp this year. I haven't heard any updates about that, but they did just get their hatchery online. There's also kind of the most reliable hatchery that's been working the longest is the University of Alaska Fairbanks hatchery down in Juneau. There are at least 2 hatcheries in Kodiak right now. The Alutiiq Pride Marine Institute also has a hatchery, and then I think there's another couple hatcheries that I am forgetting at the moment.
Awesome. Thank you so much, Angela.
Yeah, of course.
Just because you might mention that makes it way more complicated for kelp farmers to get their seeds free.
Oh.
They have to collect the kelp seed here, get it to the hatchery in Seward or Kodiak, and then get it back.
Yeah, so Kristen in the audience just reminded me to mention that, but yeah, if they aren't growing it here, it is a lot more complicated for farmers in Cordova to source their seed string, just because then you have to pack it up, ship it, like Alaska air cargo it elsewhere, and then get it sent back again via Alaska air cargo, and it's just a lot more process. And basically, the longer that kelp is kind of like in that holding stage and being shipped, more things could potentially go wrong with it. So it is really helpful for farmers in Cordova to have a spot that's in town where it's just a 5-minute drive from the docks to get their kelp to the hatchery.